Just going to introduce, got my introduction here. I'm just going to introduce Keyron now, who's from Birkbeck University, London. So he's going to talk about the field analogues. Mars is thought to have been habitable at some point, and there have been a recent discovery of organic matter associated with reduced mineral phases has stimulated new interest in the possibility of a Martian biosphere. So these materials are being sampled and will eventually return to Earth. So until the Mars sample return, they're developing a hypothesis about Mars' early habitable history requires harnessing and such settings are called planetary field analogues. So Keyron will be discussing the importance of these planetary field analogues, and in that concept, he has been sampling rocks from the Torridon Group in northwest Scotland. Keyron, in 2019 to 2020, Keyron was a research assistant at CNRS Orléans and the University of Bologna. From 21 to 24, he was Aurora Research Fellow at the Natural History Museum, London, and is now a lecturer in planetary exploration at Birkbeck. He is one of the 5 European scientists in the Mars 2020 science team. The mission is considered the first step for the ultimate Martian challenge, the Mars Sample Return Campaign. Mars has been part of his PhD research and has also worked closely with ESA's ExoMars rover team during the last 3 years. As a scientist for Mars 2020, Keyron will help guide the selection of samples for return to Earth and prepare for their study in laboratories here on Earth. Keyron received a grant from the Society here towards his recent research, which is on the Torridon. So I'll now hand you over to Keyron, who will talk about the Torridon Group, planetary field analogues, and the search for life on Mars. Life on Mars is the interesting part.
Interesting part. Thank you very much for your kind introduction, and thank you all for turning up on a Thursday evening to hear about a few wide-ranging topics as the title of the presentation implies. So we'll begin by talking about how we can use sites on Earth as planetary field analogues, which is to say that they have some characteristics that make them suitable for better understanding what we see on other planetary bodies. In this case, I am someone who mostly focuses on Mars, and so we will focus in the evening on the potential analogy for certain Martian environments of some sedimentary horizons not so far away from where we are at the moment. I'll continue through the evening thinking about how we can use some of the fossils we see in these sedimentary sequences as a tool for understanding possible fossils that may have been preserved on Mars early in its history, when we know that Mars hosted stable bodies of water, a stable long-term atmosphere. It really wasn't the inhospitable place we know it as at present. So I'll end the evening talking about how we might use some of the materials we study here on Earth to prepare in the future for Mars sample return. So in the course of the next decade or two, it really depends on who's in power in the White House, we will hopefully get some materials back from Mars. We will study these materials using sophisticated instrumentation here in laboratories on Earth, and hopefully answer the question that, as we've indicated, is perhaps one of the most interesting things we can talk about this evening, is about the possibility of a biosphere on Mars. And so I'll split the talk up into these three chapters. First of all, how we can use the Torridonian as a planetary field analog for the Noachian, Hesperian Mars. Now, the Noachian and the Hesperian are the two earliest periods of Mars history. So that will take us from approximately 4.5 billion years ago through to about 3 or 3.1 billion years ago. So this is broadly comparable with the Hadean and the early to mid-Archean here on Earth. As was mentioned a few months ago, we had a potential discovery of what's been called a potential biosignature, a potential trace of life at the surface of Mars in the form of some unusual spatial associations between organic materials, which, as we know, are the building blocks of life as we know it, but also unusual mineralogies which have formed under particular reaction conditions, and this combination can hopefully tell us something about how carbon cycling, how organic matter production, and how potentially biology would have acted billions of years ago at the Martian surface. And once again, we have advantageous examples from the Torridon Group or from the Bay of Store group, in fact, in the Torridonian sequences, that can again be made use of in understanding these sequences. At the end, I will talk about some specific analyses of Torridon Group microfossils. So these are individual cellular microfossils. I will talk about how we can study them at the micro scale, at the nanoscale, and at the atomic scale. So this is a subject of active research, and I will show you some data that we only got about 3 or 4 days ago. So you'll be the first to see some of the new data. So the first part is about the Torridon as a planetary field analog environment. We see Mars today, and it looks like this. It is a cold world. It is a dry world. It is a barren and inhospitable planet. But it is also a very ancient world. Here on Earth, a majority of the rocks are only hundreds of millions of years old. But if we go to Mars, the average age of the materials you see on the screen here are 3 to 3.5 billion years. So this is a glimpse into the earliest history of the solar system. And in particular, Mars is the planet that has certain characteristics indicating in the past it had an environment similar to that of the early Earth. It was a habitable planet. Now, at the moment, we only know a single planet that is habitable, and it's the reason we're able to have this lecture. Hopefully, in the future, we will find that we are not alone, and this will have real significance for how life evolves on planetary bodies, how different planets can have different destinies in terms of their habitability, and in terms of their possibilities for having geochemical cycling and a fossil record. On the screen here is an approximate time scale of what's gone on in the history of Mars. So you see at the top, for reference here, you have the geological time scale of Earth. Here below it is the geological time scale of Mars. And you can see that there are many similar processes. We have the formation of a stable crust. We have the formation of volcanic provinces. We have impact events. So these are planetary scale impact events by impactors up to tens of kilometers in diameter. We have also here the alteration of the surface by fluvial activity, aqueous activity. We have the development of river channels, of stable bodies of water, possibly of a planet encircling sea. And we have also the formation, therefore, of many mineral phases which are very common to us here on Earth, very well known. So if I put the green box onto the screen here, this period of Mars history, when all the exciting things were happening, when it was a geodynamically active planet, an environmentally active planet. This is the period we really care about when we think about habitability at the surface of Mars, so more than about 3 billion years ago. I mentioned that this was the time in which we think we had habitability. Here we have an idea of how habitability may have changed over time. So the blue indicator here is the habitable zone. So in the past, we think at the surface of Mars, we have the possibility for habitability. Over time, as environmental conditions at the surface have degraded, the habitable realm in blue has moved below the surface, possibly to several kilometers below the surface. It is possible that there is still a biosphere several kilometers below the surface on Mars. Simply, we cannot see it. We cannot see its effects. It doesn't have a measurable influence on the atmosphere of the planet. So it is possible that there is still a minor biosphere here, and I've put the image of the living cells on to show that that might be the case. Instead, what we see today probably are the remains of organisms, if indeed we ever had a biosphere at the surface of Mars, which should be preserved at the planet's surface. Geodynamic activity at the surface of Mars has been extremely minimal for the last 3 billion years, and so the planet's surface is really rather frozen in time. What we see now is exactly how it was left 3 or 3.5 billion years ago. And the sorts of environments we usually search for are paleolacustrine environments. So these are ancient lake deposits. And you can see here an artist's impression of a Jezero crater some 3.5 or 4 billion years ago, when it was thought to have been habitable. So you have water running in here through an inlet channel, you have water exiting here through an outlet channel, and you have a dynamic lacustrine environment. These are not the only lake environments we've explored on Earth. On Mars, since 2012, we've also had the Curiosity rover that has been exploring now for almost a decade and a half, and a reasonably similar paleolacustrine environment elsewhere. So this is Gale crater, and you see some of the beautiful sedimentary formations here. And Gale crater is also a very interesting environment. We've seen organic materials, we've seen minerals which tell us about the aqueous alteration histories. There's even a rock sample at Gale crater called Glasgow, which has been analyzed and has been found to be reasonably rich in organic materials. And so here you can see a graph showing organic detections by an instrument aboard the Curiosity rover called Sample Analysis at Mars, SAM. And the SAM instrument every few months analyzes some scooped sediment or some crushed rock samples and attempts to determine what sorts of organic materials might be present in it. And so we build up an image of organic complexity through the early history of Mars. Back to Jezero crater, because that's the one I know best. It's the one I'm able to talk to you about in more detail. So in particular with the Perseverance mission, we are focusing on one part of the crater. It's this part of the crater over here at the inlet channel. And as you see at the top right-hand side of the screen here, we have this wonderfully developed delta similar to what you might see at the Mississippi on Earth. And this is a sedimentary deposit. It's a sedimentary deposit that formed when sediments were deposited into a standing body of water. So it is a sedimentary record of habitable conditions on Mars. Here's the location at the boundary between the northern and the southern hemispheres of Mars, and here's a closer image of the delta. So you can see some of the things we'll return to here. We have the Jezero crater floor. So these are the igneous basalts and olivine cumulates of the crater floor. Here, of course, we have our sedimentary delta, which is fed by a river channel called Neretva Vallis, now a dry river channel filled with interesting sedimentary rocks. More on that in a short while. We have also some extremely ancient igneous terrain outside of Jezero craters. This is what we call the Noachian igneous basement. So at the time we're giving this talk, the rover is somewhere here. It's exited Jezero crater after approximately 4 years of examining the rocks of the crater floor and the rocks of the delta into the igneous basement. So these are some of the oldest rocks we've encountered so far. Some of these rocks may be more than 4 billion years old. They are rocks of an age that it is quite difficult to have access to here on Earth. So that's a part of the importance of studying these rocks also. Around the edge of the lake, we also have a sequence of carbonates, a sequence of lakeshore carbonates, and these are magnesium and iron-bearing carbonates. So things like a magnesite and anchorite, which are deposited dominantly around this side of Jezero crater. And to explore the crater, we have our immobile field geologist, who has many of the same capabilities as a human in the field, and some that we don't have, like X-ray vision. So aboard the rover, we have a number of instruments. At the top here, you have Mastcam Z. And Mastcam Z is the panoramic camera. All of those beautiful images you see of the Martian landscape are taken by Mastcam Z. Adjacent to that, we have SuperCam, and SuperCam is often considered a kind of Swiss Army knife of geological analysis. So it has a remote micro imager. So if I want to focus on something at a great distance, I can focus on a small region at a great distance. It's a kind of telescopic lens. It also has a few geochemical instruments aboard, helping us to understand what the chemistry of the rock targets are. At the back of the rover, we have a subsurface radar. So this is helping us to reconstruct the geometry of the Martian environment in three dimensions, and passively, as the rover moves around the surface, it takes an analysis of the subsurface every few days, and this is helping to build up an image of the subsurface geology, of the geometry of the landing site. On the end of the rover arm, and I'll come back to these quite often, we have two instruments. At the top here we have Sherlock and Watson. So just as they solved crimes in Victorian England, so too are they doing good geological detective work at the surface of Mars. So Watson is a high magnification camera. It's able to image things down to a size of about 100 or 200 microns. So imaging individual minerals, individual crystals, individual aspects of the rock microtexture on Mars. And then Sherlock and Pixel are two geochemical instruments. So they are mapping the distribution of elements in the rock in the case of Pixel, and the distribution of specific mineral and organic phases in the case of Sherlock. So when we seek to identify organic materials in the rocks, we use the characteristics of the interaction between the ultraviolet Raman spectrometer aboard Sherlock, and we can match up these detections in space with the images we get from Watson. So we can say, on this one particular 100 micron pixel of Mars, we have identified organic material. And so this is one of the strengths of the rover, is that it is able to see spatially what the organic, it is able to see spatially where the organic materials are distributed. So that's a real strength of the rover. There are two other instruments aboard also. We have a weather station, which takes measurements every day, and we have an instrument called MOXIE. MOXIE is a technology demonstration. It produces oxygen from Martian CO2, atmospheric CO2. And the objective here is that in the future we should have humans at the surface of at the surface of Mars. We can decide how we feel about that ourselves, but we can if we have humans at the surface of Mars, we need oxygen. We need free oxygen. We cannot take all the oxygen we need with us. And so this is a very small instrument. It's probably the size of this box on the desk. But the idea is that in the future, this could be made at an industrial scale for producing sufficient oxygen to keep humans alive for sustained periods at the Martian surface. And so many of the rock types we see at the surface of Mars are basalts. And so here are some smooth and some wind-fluted vesicular basalts at the Martian planetary surface. These are very much of interest for questions of geochronology, understanding how old certain features on the Martian surface are by dating specific minerals in these rock samples. So in the future, having them back here on Earth, we'll be able to date them in the laboratory using uranium, thorium, lead systems, and we should get an idea of precisely how old certain parts of the Martian surface are. At the moment, we usually do this by estimating the densities of craters across the Martian surface. Here we could get an absolute age. We know that this part of the Martian surface is 3.68 billion years old, for example. But we also have an abundance of sedimentary materials. Here in the distance, you can see some sedimentary hills, and so these are dominated by phyllosilicates, mostly by montmorillonites and various smectites and possibly illite. So these are some of the delta deposits of the crater. But there are some that are far more impressive. Here you can see a delta remnant, and you can see the characteristic top set, foreset, bottom set associations, sedimentary associations in the delta. So just as we see these kinds of sedimentary associations on Earth, so too can we see them on Mars, and these help us to understand some of the characteristics of sedimentary deposition. It's quite hard to see here, but there are also boulders and pebbles mixed in with these sedimentary horizons. So this was not always a quiescent environment. We also had periods of extremely high energy deposition. Here are some of the cliffs of the delta front. So these are approximately 30 meter high cliffs. These also provide us with abundant evidence for the kind of sedimentary processes that would have dominated Jezero crater during its habitable phase. And again, we have collected samples of these materials. So aboard the rover, we have samples in sample tubes, and they will hopefully in the future be brought back to Earth. Here is a particularly impressive image, however, of the Jezero delta front. Here you can see the wonderfully laminated layered strata of the Jezero delta front. These materials were indeed one of the major focuses of the Mars 2020 mission. This is why we designed the mission with these objectives in mind, are to see what we can identify in the sedimentary rocks of the Jezero delta. Here are a few close-up images of these rocks, and indeed you have a real diversity of sedimentary materials. We have some very fine-grained mudstones. Now we know here on Earth that mudstones are extremely good at preserving large quantities of organic materials. We know that also from Gale crater. All of the organic detections by Curiosity have been in Martian mudstones. For example, Glasgow is a Martian mudstone. We have here at the bottom sandstones, and so sandstones, higher energy environments, but then again here we have conglomerates. So a range of energy settings through the course of the delta. Most of the geochemistry we've seen of particular interest again has been in the mudstones. And we can understand up close what the chemistry of these particular rocks are. So these images on the screen elemental maps taken by the pixel instrument. So you put the instrument directly onto the rock target, and over the course of a few hours, it analyzes the X-ray fluorescence of each of these pixels, 100 micron pixels, and builds up an image of the concentrations of particular elements in these Martian horizons. And so we can see in the sedimentary rocks a range of very familiar phases. We have calcium sulfates. We have iron magnesium carbonates. We have a generic siliciclastic phase, a mudstone. We have some more unusual minerals, greigite. It's an iron sulfide, but then also some igneous minerals mixed in, olivine. So we can get a real idea of the mineral diversity of the depositional system. Here are a few more images here of some altered carbonates. So again, here we can drop the pixel map onto the rock target. Each of these pixels is a spectrum, and from that we can assign mineral groups. So we can tell the difference between carbonates, between serpentines or other aqueously altered minerals, and for example silica. So again, we know that silica here on Earth is extremely good at preserving organic material. Many of the most ancient traces of organisms on Earth, 3 or 3.5 billion years old, from say Australia or South Africa, these tend to be in chert, a silica-rich rock, and so seeing silica-rich phases on Mars, well, that's also the sort of place we can search for the organic remnants of a potential biosphere, and we can couple these analyses with a visible and near infrared spectroscopy. So again, we have our data sets from Mars at the top here, and we can see particular absorption characteristics. We can see particular fingerprints of individual minerals. We can compare these with pure mineral spectra here on Earth, which we gather in the laboratory. We can compare with what we see on Mars and begin to get an idea of the range of mineral phases present in these rocks. And these, of course, tell us about the kinds of environments in which they formed. Are they alkaline environments? Are they acidic environments? Are they high temperature? Are they clement environments? And all of this, again, is important for identifying potential traces of the biosphere. We have these wonderful sequences on Mars, wonderful sedimentary sequences. We have a very similar sequences here on Earth. They have similarities in terms of their depositional environments. They have similarities in terms of their mineralogy. They have similarities in terms of their current characteristics of the energy of deposition. So you can see a beautiful rippled bed here. You can see some convoluted sedimentary laminations here. So these are the kinds of environments we think about studying, with similarities in terms of their mode of deposition, similarities in terms of their mineralogy, in terms of their chemistry. All of these factors help to make a particular setting here on Earth useful for interpreting data sets from unknown or poorly known places on Mars. And it's important to note that there is no perfect Mars analog here on Earth. We have a planet which is hopelessly polluted by the biosphere. We all act upon it every day, and so all of these environments here on Earth are affected by us, are affected by the diversity of our biosphere. So we have to choose which characteristics we think of as being analogous to environments we see on Mars, and mineralogy and depositional environment are two of the real key parameters if you wish to understand in which kinds of settings life might have arisen on Mars. That's just one locality in the Torridonian. Here we also have Upper Diabag. So maybe some of you know this wonderful road cut in the upper Diabeg area. Here we have, you can just about see it here, a variation between the more oxidized mudstones and the more reduced mudstones in greenish gray here. So these again are similar associations to what we see in mudstones on Mars. So we have chemical variations or chemical processes which have some analogy again to what we see on Mars. So far, I've been talking mostly about Mars 2020, but in the bottom corner here, I show you an image from a few weeks ago. This is not actually Mars 2020. This is a simulator of the camera aboard the ExoMars rover, aboard the European Space Agency's ExoMars rover, which is in a laboratory about half an hour from where I live. And so I said, I have these beautiful samples from Scotland. May I bring them down and image them? And they very kindly granted us access to a few days of instrument time at the lab, and we were able to see how we could image. So you can't really see it in this image here, but there are microbial mats or microbial biofilms, trace of a biosphere, in these particular rock samples which were collected at this outcrop here. And so I hope that gives a sort of tangible link how the rock samples you may know quite well from the field are able to be integrated into preparations for planetary exploration. That's Upper Diabeg. We also have Eilean Floodday, so this is off the coast of Raasay. I'll come back to this a bit more in the second part, but there are these unusual phosphate nodules in the sedimentary horizons at Floodday, and these are superb repositories of biosignatures. Every one of these nodules is chock full of hundreds or thousands of microfossils, cellular microfossils. And so these are some of the materials we are studying in order to understand how we can identify and characterize and give identities to the organisms that are preserved in these materials. Also, you have at Clactol and the Bay of Storr what are potentially Britain's earliest traces of the biosphere, and these come in the form, if you go in the bay here, you can go over to this side close to the Clactol, and you can see these wonderful wrinkly laminations. There is a hypothesis that these are the remains of a microbial mats, and that these archive their interactions with sedimentary materials. If you go over to this side, just at the foot of this small cliff face here, you'll also find some other unusual materials, and these are iron replaced horizons, which have, as you see, an extremely complex internal lamination. And again, there is a hypothesis that these are ancient stromatolites. The jury is a bit out on these at the moment. Certain people support a biological origin. Others say no, these are entirely sedimentary phenomena, and there are no traces of organisms in these. So that's something of an open research question at the moment. But I think again tells us something about here in Scotland are some of potentially the most interesting rocks in the world for understanding potential biospheres on the red planet. So coming back to astrobiology on Mars, all of the environments I've shown you there, planetary field analogues, have some sort of relevance to various parts of the Jezero system. And when we think about astrobiology, the search for and the understanding of the distribution and the characteristics of a biosphere through the universe, we think of a number of other factors. We have to think about energy. How did these organisms survive? We have to think about the raw materials. Have they got the carbon, hydrogen, oxygen, and nitrogen and phosphorus that they require to metabolize and to succeed in environments? Are these environments favorable, or are they particularly challenging for the survivability of the biosphere? And because we are tremendously Earth-centric in our approach, is there water? We only understand how a biosphere is able to function if the solvent is water. Is that necessarily the case on other planets in the solar system, where the geochemistry and the bulk chemistry of these bodies is different? The caveat we have is that we don't know better, and water is the only solvent we understand in terms of the search for life. So you have habitability. Then habitability feeds into how we define exploration targets on other planetary bodies, but also here on Earth. We have habitability as one important factor. We have, of course, detection. Are we able to identify biospheres? If, for example, life on Mars is arsenic-based or silica-based, we don't know what we're looking for, and so we may walk straight past it. We may analyze it, but we may not realize it's a trace of a biosphere. Now, I think that those other biochemistries aren't particularly probable on Mars, but they become more probable as you go to other places in the solar system, thinking about the icy worlds, for example. Now, this detection goes hand in hand with technology. On Mars, we have certain instruments aboard the rover, but they tend to be far less powerful than the instruments we have here on Earth. This is because we have a shortage of space, we have a shortage of energy, we have a shortage of sustained power. You cannot keep operating an electron microscope at the surface of Mars, although it would be extremely handy if we could have one. So you are restricted also by the instrumentation you have on Mars. You are restricted by the technology. Here on Earth, we are limited largely only by our own imagination. And I'll talk a bit more about that in the third part of the talk. And then, of course, we have preservation and taphonomy. How are things preserved? 99% of shells do not enter into the fossil record, and shells are extremely resilient. Here we are searching for fossilized bacteria. They're extremely fragile. If they're made of organic material, the odds are seriously against any of these entering into the fossil record. And so you really need exceptional preservational conditions, exceptional taphonomy. You put all of these things together and you get your exploration target. Now, some of these exploration targets are not as one might expect them to appear on other planetary surfaces. A few months ago, there was a paper published in the journal Nature, and it was all about this particular sample and the rocks that it was collected from. This was discovered in June 2024, and the headline was "Perseverance Finds an Intriguing Rock." And that's a kind way of saying we don't know what this is, so we're going to say we find it very interesting. And so here is our intriguing rock. It's a rock sample called Sapphire Canyon. So here on the screen you see a view which is looking at the top of a small core sample which has been collected by the rover, and the core sample is about the size of my finger. It's about a 1.5 centimeters across, about 8 centimeters long. And this came from another fluvial environment close to Jezero crater. So here, you remember we were at the delta here. That's our Jezero delta. Here is the river channel. So we will now talk about this particular inlet region of the river channel. This is a river channel called Neretva Vallis. And within the Neretva Vallis area, you may see some pale rocks here at the right-hand side of the screen. So these are these pale rocks here that we can see in the satellite image. And it's a rock formation called Bright Angel, apparently because it looks like a pair of wings. Maybe you can see it, maybe you can't. It really depends on the day for me. And at these bright rocks, we have these flat pavestone like outcrops. Here you can see the shadow of the rover as it drives across them. And we've got these unusual layered sedimentary materials. Here, the scale bar is approximately 10 centimeters. And we focus in on this region here because in this region we have these unusual spotted textures, and you may have seen that these were called leopard spots in the news. And so this is a granular rock. It's a very fine-grained rock. It's a mudstone, and it's cross-cut by these unusual mineralized veins. We again put pixel onto the rock, so you can see here a map of the rock has been obtained, and these again are the chemical maps of the material. You can see in pink we have our mudstone. In the background, our phyllosilicate rich mudstone, and we have these unusual poppy seed features which have core that I've marked out in purple here, and a rim that I've marked out in green, and these features, informally termed leopard spots, are the particularly interesting features we've seen. They appear to have formed in situ during and after the deposition of these horizons of mudstones. And the interesting thing is that they are associated with organic material. So these are the kinds of data sets we get back from the Sherlock instrument. It's a kind of spectral data set. And you can see a feature is marked out here, which is called the G-band. And the G-band is a diagnostic feature of organic materials. So having seen this feature present in the rocks, we know that there is a small quantity of organic materials here. You can see what the background signature is. It's a very weak potential enrichment here. Then you can see in some of the other signatures that this becomes a much stronger signature. And so we have an unusual rock, an intriguing rock indeed. It is rich in a number of unusual minerals, iron phosphates, for example. It's a mineral called vivianite in the leopard spots. These are embedded in a mudstone matrix, interlayered in green with calcium sulfate in the veins. And this particular organic mineral association is unusual. We have iron phosphates and we have iron sulfides. These are reduced iron minerals. So these are iron 2+ bearing minerals. They are in an oxidized environment. So there was clearly some transfer of redox chemistry. There's evidently an expense of energy in order to produce these unusual leopard spot textures. And so this is the reason it has been put forward as a potential biosignature. You have the mixing of the reactants in the rocks. You need to reduce both iron and sulfur into these unusual minerals in the purples and blues in these maps. You need to oxidize organic materials. So this is a very complex reaction between minerals and organic materials, all of which have been demonstrated to be there. And here on Earth, these are reactions which take place under biological activity. In the paper, there is a sentence here: Bright Angel, the rock outcrop I showed you a while ago, is a habitable environment with textures, chemical and mineral characteristics, and organic signatures that warrant a consideration as potential biosignatures. I mentioned earlier on that aboard the rover, we only have a certain range of instruments. We don't have the power aboard the rover to have the more powerful instruments we have here on Earth. And so the essential next step becomes Mars sample return. We need to study them here using organic geochemistry at extremely high sensitivities on Earth. And this will hopefully enable us to revise that description of potential biosignatures into either, yes, these are evidence for life on Mars, or no, they are not. And both of these outcomes are interesting. If we have evidence for life on Mars, we show that we are not alone in the solar system and in the universe. We have other biospheres on other planetary bodies which were habitable at some point in their history, and I think the implications of that are clear. If we find that these are not biosignatures, well, that's also tremendously interesting. It means that you can have a perfectly habitable planet, very much like Earth in its early history. It has an atmosphere, it has water, it has all the ingredients and all the nutrients required for life, and it doesn't happen. And that means that there's something very special about Earth. So in both of these instances, it's a very interesting outcome. We cannot know the answer until the rocks are brought back here. These are not the only such textures we've seen. About 500 meters away, we see these unusual green reduction spots. Tragically, these were not collected, and that was due to some pressures from a NASA administration above, who said you have to keep the rover moving. And so unfortunately, these were observed, but they were not collected. We are approaching the end of the mission, and we still have 4 sample tubes remaining. There is a possibility that we will collect these in the future. Again, we can think about planetary field analogues. If you go back to the Bay of Store, you have just below the Stackfader member a sequence of reduction spots. It's just around here, behind this small hillock. And these reduction spots, again, are green reduction spots with unusual dark cores. Here is an electron microscope image of tiny crystals of pyrite, of framboids of pyrite, in the middle of these materials. These reduction spots seem to have formed due to biological activity. This is rather promising for the sorts of things we see on Mars, that there might be a similar biological activity present there. And although the images aren't particularly ideal, they are rather pixelated, you can see many similar characteristics. We have a dark core here on Earth, a dark core here on Mars. We have a pale green-gray exterior, a pale green-gray exterior in an oxidized mudstone, And the parallel is extremely strong between these materials in the Torridonian again and materials we have on Mars. The examples on Earth are probably formed by the terrestrial biosphere. So what does that mean for the examples on Mars? In the 10 minutes I think I have left, is that okay? Approximately, yes. That's a yes. I'll talk about in a bit more detail some of the Torridon microfossils. And here I think about how we can look into the future when we have samples back here on Mars, back here on Earth. We think that they contain a potential biosignatures. What can we do with them? We'll go back to a place I mentioned earlier on, which is Eilean-Flade. You've got these phosphate nodules, black phosphate nodules, and these contain hundreds and thousands of microfossils. We can make thin sections out of these rocks, and so here's a thin section through a nodule very similar to this one. We can look inside, and they are indeed full of organic microfossils. Over here, arrowed in black, we have small fossilized cyanobacteria. Here, arrowed in white, we have these larger coccoidal or spheroidal organisms, which have been interpreted as potential eukaryotic organisms. Here, arrowed in green, we have an unusual disc-shaped microfossil, several tens of microns in size, so that's quite a large microfossil. And then over here, maybe you can see arrowed in red, we have a very faintly preserved filament. So there are a range of cellular morphologies preserved here. So we are clearly at the center, or close to the center of our exploration target diagram. We're in a habitable place, and the proof is we're here. We have something which has been exceptionally well preserved, and the proof is that they are there. We have something that we can detect, we can see them, but have we got the technology to identify these and characterize really what they are? So on the screen here, you can see a few scanning electron microscopy images and elemental maps. And this was work kindly supported by the grant received from the Society. So you can see here an electron image of an individual fossil cell. Here we are down to only a fraction of a size of a human hair in dimensions. You can see in black here we have the fossil cell wall, so that's shown up in the map of carbon. But in the interior of the fossil, we can see these unusual silicate minerals. We can see more predictable silicate minerals. So these are aluminium and potassium clays. And we can see in the exterior here, we have calcium phosphate, microcrystalline calcium phosphate, which encapsulates our microfossil. We can go in a bit more detail here, and we can characterize the organic material. So at the left-hand side here, you see some maps, again at the microscale, of the distribution of carbon and the distribution of a nitrogen in these fossils. So these are organic materials made of indeed many of the same elements that make up all known organisms. So these were some analyses performed with some colleagues at the Korea Basic Science Institute in South Korea. And then again here, we know that we have carbon and nitrogen, so we can begin calculating some of the isotopic ratios of these elements. And this again is ongoing work, which was again supported by the grant received from the Society. So we have here, these are reasonably strongly negative carbon isotope fractionation. So that is to say these organisms are enriched in isotopically light carbon, but isotopically heavy nitrogen. Now, this is quite interesting because it tells us something about the kinds of metabolisms which these organisms might have been using. How are they harnessing carbon and nitrogen in the atmosphere, in the environment, in order to produce and conserve energy? And it seems that they are producing it via carbon and nitrogen fixation. So these are metabolisms commonly used by cyanobacteria, which are some of the main architects of oxygen in the atmosphere through Earth's history. So we can find out more about how these are preserved and what we can find out about these organisms at the organismal scale. The images you see on the screen here are maps collected with an infrared spectroscopy. So we target individual microfossil cells, and again you can see the green is the carbon of the cell wall. of the microfossil cell. But we can see it isn't only made of a plain old carbon. We have a number of interesting carbon groups. We have carbon bound to oxygen. If I go over the page, we have carbon bound to nitrogen, carbon potentially bound to hydrogen. So these are all the elements, again, that make up organisms here on Earth. And we can see, so in these Torridon microfossils, 1 billion years old, they preserve an archive of their own compositions, and they tell us something about their biochemistry, something about their style of existing a billion years ago. There are a whole host of spectra here, which I won't go into detail on, but that's the demonstration of what's in the maps at the left-hand side. So organic materials in association with phosphates and in association with silicate minerals are able to archive something about their own biochemistry over billions of years, which is what we need on Mars also. We can go down to a very, very fine scale also. So you notice now that we're down to about a micron. We can extract tiny needles, tiny subsamples out of the cell walls of these materials. So this is using an ion beam. We have to cut atom by atom through the fossil, and we eventually cut out a small needle of material. And again, we can see what are they made of. So that's the fossil. Here's in the center is the anatomy of the fossil once we've cut it open, and then we can extract out of a tiny region of the fossil a needle of material. We can see what this is made out of. So this is a technique called atom probe tomography. I discovered recently that you'll be getting one at the University of Glasgow soon, which is extremely exciting because here in the UK we have only perhaps 2 or 3 of these instruments, and they're dominantly in a material sciences departments. So the fact that you'll be getting one here for a geoscience applications is extremely exciting. So in these element maps at the right hand side of the screen, each of the individual dots, each of the individual points here is a single atom. So we cut our way through this this needle atom by atom, and we can see again the distribution of the mineral phases and the organic phases. So up here at the top we have carbon. So that's a tiny fragment of the cell wall of the microfossil CW here. We have the matrix, which is again phosphorus and calcium. So that's a microcrystalline apatite. It's the M here. And this is the interior of the fossil, intracellular region. And this is rich in silicon, oxygen, plus aluminium and potassium and some of the other minerals I talked and some of the other elements I talked about a few minutes ago. And so we can see at the nanoscale, or indeed at the atomic scale, how these organic materials are preserved. Similar associations exist at Jezero crater. These pixels here show the fluorescence signatures of organic material which have been detected by the Sherlock instrument on the arm of the rover, and they are associated with minerals we have spoken about here, amorphous silicates, which are potentially very fine-grained clay minerals, phosphates, which again we've seen. So that small red square here is the same as the red square here. Here are the amorphous silicate minerals detected. Here are the phosphate minerals detected. So again, an association of organic materials, minerals, which we see on Mars and that we can see again here in the Torridon Group microfossils. So the punchline is that these are kinds of analogues, planetary field analogues at a number of scales. They are analogues at the environmental scale. They are analogues at the mineralogical scale, but they could also be analogues down to the atomic scale. And that makes them, I think, an exceptionally valuable national resource, unique in the country and probably unique in the world as being this kind of planetary field analog at all scales. Of course, what we need is to have these materials back here on Earth. Here is a sample tube that's been dropped at the surface of Mars, and this is a very important first step. Once the rover had deposited the first sample tube, the planning for Mars sample return is able to begin. You need samples on the surface so that you can pick them up in a future mission and collect them. So Mars sample return becomes a reality when this sample is at the surface of the planet. Obviously now what Mars sample return requires is, first of all, support from politicians. Secondly, money allocated from the US federal budget. But at the base of all that is enthusiasm and encouragement from scientists and the public, who at the end are the people who either fund or will do this kind of work. On the screen here is the Hall of Fame of almost all of the samples which have been collected so far. It's a few months out of date. They haven't updated the bottom line. Here is a sapphire canyon, which we spoke about a few moments ago. All of the ones that I've encircled with red, these are the ones that have clear analogues somewhere in the Torridonia. So that accounts for more than 30% of the total sample cache. We have access to those kinds of analog materials here. And yes, what sorts of organisms might we be expecting to think about? I will end on a fun note here. So at the time of the discovery of the organic materials and the leopard spots, I was contacted by a journalist from a well-known tabloid, and he said to me, what sorts of organisms do you think we might find? And I said, I think they're going to be similar to what we see in a microbial ecosystems here on Earth. Pond scum, he said. Well, somewhat, but we prefer to think of them as microbial ecosystems. You know, seems a little more dignified if we give them that name. And he said, okay, so we're going to try to create some artwork of the sorts of organisms we might expect to find on Mars. And he showed me this one. I said, that's actually really nice. That's the kind of thing that we could be expected to find. The article came out the following morning. It was accompanied by 2 images. It was accompanied by that and this. I can assure you, if this organism were living on Mars at any time in its history, it would be having a thoroughly miserable time of it. Thank you very much for your attention, and and again, thank you very much both for the award of a grant to indeed further many of the fundamental parts of this work, some of which are still ongoing, particularly understanding some aspects of the rare earth element, the rare earth element chemistry of these rocks, and how that can feed into our understanding of the paleo environment, but then also for the invitation to come here and speak with you. I hope that was interesting, and I'll be happy to take any of your questions. Thank you very much.